Hello everyone, myself Dr. M. Sumitra. Now I am discuss about terms related to UV visible spectrometry. The first one is chromophore. Chromophore means the covalently and unsaturated group responsible for the absorption of ultraviolet or visible radiation. It may or may not impart color to the compound. Generally, chromophore means the compound containing the unsaturated group or functional group will be act as a chromophore. A compound which contains chromophore is called as chromogen. The unsaturated linkages such as carbon-carbon double bond and nitrogen double bond nitrogen, it contains the loosely bounded electrons. We are pass the radiation over to this type of group, the electronic transition will be happen and it will be responsible for the absorption of UV and visible radiation. Generally, during that process, the loosely bounded electrons moving to the electronic transition and it produce the absorption in that UV and visible region. The example for the chromophore is ethylene, acetylene, carbonyl, acids, esters, and nitrile groups. Next, we move on to the types of chromophore. Generally, the chromophore are classified into two types. The classification will be done by according to the electronic transition in the bondings. The first one is the electrons undergo transition in between pi to pi star bonding. In that method, bonding orbital move on to the pi star anti-bonding orbital in a molecule. These are also called as dependent chromophore, as more than one chromophore is required to impart color to the chromogen. The example for the chromophore is ethylene and acetylene group. The another type is second type chromophore. In this type, both pi bond orbitals and non-bonding electrons undergo the transition in a molecule, that is pi bonding orbital electrons to pi star anti-bonding orbitals and non-bonding electrons to pi star bonding transition in a molecule. These are also called as independent chromophores. It will be occur in the single chromophore and it is sufficient to impart color to the chromogen. These are all the example for the second type chromophore, that is azo group, carbonyl group, nitrile group and nitro compounds. The next important term is oxochrome. Oxochrome means it coming under the saturated group in the molecule. In that oxochromine, it is a saturated group which consists of one or more pairs of non-bonded electrons. These groups, when attached to chromophore, helps in altering the wavelength. That is, by increasing the intensity of absorption and shifting the absorption maximum towards the longer wavelength. Actually, the oxochromine, it enhances the absorption of your radiation. These are all the examples for the oxochrome, such as hydroxyl group, amine group, amide group, and esters. The next important term is isobustic point. Isobustic point is a specific wavelength at which two chemical species have the same absorptivity. Such a point corresponds to a wavelength or an isobustic plot at which the absorption spectra of two species cross each other. So, as per the definition, isobustic point mean is a point at which the more than two or more species will be produce the absorptivity at the same point. That point will be called as a isobastic point. In that spectrum, the wavelength are plotted in the x-axis and absorbance will be plotted in the y-axis. If you are checking this spectrum, the isobastic point at which the three compounds are crosses each other. So from this spectrum, we can understand the isobastic point in that UV spectra. So next will be the icebreaker section. That is, from these quizzes, we can brush up your mind. By using this link and the passcode, we play the quizzes. These are all the reference for my presentation. Thank you.